Hi everyone, this is Mr. Hall and this is a really quick video about enzymes. This will be the first of many videos, hopefully, which I'll be making about enzymes, because enzymes is quite tricky and there's quite a lot that you need to know, particularly for like the GCSE. If you do an AQA or something, you need to know about how they work, you need to know about required labs, stuff like that. So hopefully this video will just be sort of like an introduction to enzymes, and then future videos will go a bit more into the details of different experiments and stuff like that. So in today's video, I'll be talking about the following. I'll talk about what the definition of an enzyme is. I'll talk about what enzymes do. I'll describe the ideal pH conditions for different enzymes in different parts of the body. And I'll also describe what happens to enzymes in different pH conditions. For this last one, I'm not going to go into massive depths about it, but I will just talk briefly about it. In future videos, I'll talk more about the active site and stuff like that. So let's get started. So digestive enzymes, they are biological catalysts. And what they do is that they break large insoluble food molecules into smaller soluble ones. Just to break down this language a little bit for you. So if it's biological, it basically exists in living things. And then the idea of a catalyst, if you've learnt this in chemistry, it's basically that it makes things happen quicker. So basically what they do is that they break down food inside living things quicker than what would normally happen if they weren't there. Now, what they do is that they break insoluble food molecules into smaller soluble ones. So if you think about an insoluble food molecule like this, because it's insoluble, it means that it won't dissolve in a solvent like water. Okay, so it won't dissolve. Now, this is a problem because if it won't dissolve, it won't go into your bloodstream, and you need it in your bloodstream so it can go to all the different cells in your body and then to help your respiration and stuff like that. So it's important that it's soluble. At the moment it's not. So what we do is we get some enzymes, which are basically like biological catalysts, like scissors. They help cut them up, and then once they're cut up, they can then break down into smaller pieces. And then when they get into smaller pieces like this, they are then soluble. And then when they're soluble, they can then go into your blood, and then they can go all around your body. Okay, so that's really important. Okay, now there are different types of enzymes that you have to know, and they work on different food types. Now the first one that we'll be talking about, which I'll mention now, you've got carbohydrase. An example of that is amylase, and that breaks carbohydrates into smaller sugars. You'll find that in the salivary glands as well as other parts of the body. You've also got protease, and protease breaks proteins down into amino acids. You tend to find that in the stomach. And then finally, you've got lipase, which breaks fats down into fatty acids and glycerol, and you find that generally in the small intestine. So that's in the small intestine. I'll just put SI. This is in the stomach. And here you'll find this in lots of places, but also in the mouth where you've got saliva. Okay. You'll notice there's obviously a pattern in the name: carbohydrase, carbohydrate, protease, protein, lipase, lipids, although we tend to call them fats more colloquially. So let's talk about carbohydrates first of all. So carbohydrates are basically long chains of identical sugar molecules, and then what you have is carbohydrates coming in, and it breaks the chemical bonds between each individual sugar molecule, and therefore it can be dissolved in your blood. Okay, so this is our long carbohydrate molecule. An example is starch, and this can get stored inside the body or inside plants even as a store of energy. And then your enzyme comes along. Remember, we call it like a biological catalyst, like a scissor. So this are carbohydrates, and they can go between each of those different bonds between each individual sugar molecule, cuts them up, and then you've got these individual sugar molecules which are now soluble. So now these are soluble, they're able to be dissolved in the bloodstream. Okay, so that's how that works. For proteins, now proteins are not made of sugars, they're made of things called amino acids. Now the thing is, they are different. These amino acids are different, and that's why they make proteins. And the enzyme called protease breaks the chemical bonds between them in order to form your individual amino acids. Okay. Now these work best in an acidic environment, which is obviously in the stomach, and that's part of the reason why your stomach contains stomach acid, which contains HCl, very low pH. So here's my amino acids, okay, and these, or my protein rather, and all these different shapes represent different amino acids, okay? Now you want those different amino acids in your body, so then you can make your own proteins, okay? So then you get protease, which is represented by the red scissor, because acidic environment, and there they are. They start cutting up those bonds, and then you have those individual amino acids. Now that is soluble, so now that can go into your bloodstream and go to different parts of your body and start making proteins and whatnot, okay? And then we've got fats. Now fats is a little bit more complicated. Now fats are complex molecules, and they're made of two things. You've got glycerol, which is like the big chain, and then you've got these fatty acids, which are these smaller chains which come off it. Now, the digestion of fats happens in 2 stages. The first thing that happens is you have bile. Now, bile is basically the stuff that you basically have in your vomit. It's not very pleasant. It's released by the gallbladder. All right. Now, the gallbladder is this small little sac which is just below your liver, right here, and that stores your bile. Now, what happens is your gallbladder releases the bile. It mixes with the food, and the fats, I should say, and then what it does is it basically breaks that fat down into smaller droplets. Now, that's an important step, because if it doesn't, then what you'll have is your fats and any water inside your digestive system not mixing, and if they don't mix, then it's not able to break the stuff down. The word for that is immiscible, okay? And if you think of like oil and water not mixing, it's similar to that. The fats just won't mix with the water. You don't get digestion happening. So it's important for it to be mixed with the bile to break it down into smaller drops. And that process is called emulsification. So one way to think about it is you've got this massive sort of blob of fat inside your digestive system. Bile comes in, and after doing its action, what you then got is lots of small droplets of fat. And then it makes it much easier for enzymes to break it down. So the enzyme for lipids or fats is lipase, and what it does, it breaks the chemical bond between the individual fatty acids and the central glycerol chain. Okay, so these work better in an alkaline environment, and this is something which I'll mention in another video, but your alkaline environment is found in your small intestine. Okay, so this is my fat molecule. Now, this thing here, this large, so these pentagons on this chain, that's glycerol, okay? And these individual things here, these are your fatty acids. So we need lipase to break down this fat molecule, which I'll represent by a blue pair of scissors, because obviously it's in an alkaline environment, and it goes here. So what it's doing, it's cutting this bond. Okay, so as soon as it's cut, you then have 2 products. You've got glycerol, which is on its own, and then you've got the individual fatty acids, which are made on the side too. Now, enzymes can only work on specific food types. So if I try to use one type of enzyme on a different type of food, it's not going to break it down. So for example, if I try to use protease on carbohydrates such as starch, not going to work. So for example, here's my long carbohydrate molecules such as starch. If I try to use protease on that, that's not going to work, because protease only works on proteins, doesn't work on carbohydrates. Now, the reason for that is due to like the shape of the active site, but that's something I'll cover in another video. So essentially what will happen here, nothing gets cut, nothing gets broken down, and then you still remain with your carbohydrate molecule. Okay? And that's it. So I hope that was helpful, and best of luck with your studies on enzymes. More videos to follow.